the way I think about it in the lab is, we want to kill the compound. We want to kill the series. You want to kill the target. You want to kill the mechanism. Because, if we can do that as well, it's great basic science. It lets people know where not to go and allows us as a collaborative, community to focus on the targets that really could impact patients.
Today, I'm excited to welcome our guest, Dr. Craig Lindsley, a cutting-edge innovator and scientist in neuroscience drug discovery. Dr. Lindsley is a leading medicinal chemist and neuroscientist with almost 3 decades of experience in drug discovery and development across industry and academia. He's a university distinguished professor of biochemistry, chemistry, and pharmacology, and the William K. Warren Jr. Chair in Medicine and executive director of the Warren Center for Neuroscience Drug Discovery. Welcome to Quantum Potential, a podcast dedicated to curiosity and discovery. My name is Cybele Raver, and I serve as the Provost of Vanderbilt University. As a leader in higher education and as a social scientist, I believe that we can transform higher education for the 21st century. At my core, I am wildly curious about other people's work, whether it's in science, the social sciences, the humanities, or the arts. This podcast gives us the opportunity to break open new ideas through discovery and creative expression. The Warren Center for Neuroscience Drug Discovery is a powerful hybrid academic and industrial drug discovery engine. And the work at the Warren Center, with Craig Lindsley leading, has advanced novel treatments for therapeutic areas around cancer, diabetes, obesity, and neurological disorders like Alzheimer's, schizophrenia, Parkinson's disease, anxiety, and pain. Dr. Lindsley is also editor-in-chief of the American Chemical Society Journal of Medicinal Chemistry, and his groundbreaking contributions have earned him induction into the American Chemical Society's Division of Medicinal Chemistry Hall of Fame, the Royal Society of Chemistry, the National Academy of Inventors, among many, many other accolades. Dr. Lindsley continues to push the boundaries of drug discovery to help people suffering from multiple medical challenges across the United States and across the world. Craig, let me begin by saying that we are so grateful for all the work that you're doing. Really exciting to have you today.
Well, thank you so much. It's great to be here.
So today we're talking about cutting-edge neuroscience research, breakthroughs that could change the way that we treat major threats to our health and well-being, like schizophrenia, Alzheimer's, and chronic pain. Craig, before we dive into your work discovering new medicines to treat those incredibly debilitating conditions, just help us understand a little bit more about the brain. Specifically, how do we think about the way in which Alzheimer's disease affects roughly 7 million people with no known cure? Many of us listening have a family member or a loved one who suffers from these terrible neurodegenerative diseases. But there's a lot we don't understand about the brain. What's going on with Alzheimer's and Parkinson's? What's actually happening in the brain?
Well, it's very complex. And it really kind of goes back even to the way we think about oncology now and personalized medicine. Everyone's Alzheimer's symptoms may appear the same, but the etiology of the disease can be very, very different. Mm-hmm. So what we do know is you see severe loss in multiple signaling pathways. We see degeneration of the brain. We see loss of brain mass, aberrant signaling. And so there's never going to be, I think, a magic bullet, one drug that's going to cure everything. And even now, as we think about clinical trials, we really have to think about palliative care and just improving cognition and keeping patients out of assisted care facilities. And so the easiest thing right now to work on is behavioral disturbances and increasing cognition and working memory by modulating different receptors that are downregulated in the disease.
Great. I mean, I think you talk a lot about positive allosteric modulators, right? These PAMs, and you also talk about negative allosteric modulators. What is that? What is a modulator in the brain? How does that work in terms of signaling?
Yeah, so a lot of times what you'll have is a receptor that's either has a loss of function or a gain of function. So we want to turn it down or turn it up. And historically, especially with a lot of families of receptors, people use what we call an agonist or an antagonist, which would directly activate or directly inhibit the receptor.
And this is a receptor on the
neurons.
Mm-hmm. And it's affecting how the neuron takes up or doesn't take up neurotransmitters like dopamine, right?
Mm-hmm. And acetylcholine and things like that. So The problem with agonists is they bind at a very conserved site across a family of receptors, so you typically get poor selectivity. So if we think about the drug that was just approved by BMS, Cobenfy, which is a muscarinic agonist, it activates all 5 subtypes. And so they have to co-administer a peripheral antagonist with it to avoid GI side effects and other problems. What we've done is we target the allosteric sites that are not conserved.
And just to slow down, what is an allosteric site?
It just means other.
Okay.
So it's a site other than where the native neurotransmitters bind. And so by doing that, we get amazing selectivity for 1 out of a family of maybe 5 different subtypes. And so we can only activate the receptor of interest, not all these others that could cause adverse effects. And then a PAM, the way to think about it is when a positive allosteric modulator binds to a receptor, it doesn't do anything on its own. But when the native agonist like dopamine or acetylcholine binds, you get a heightened response. And so it's more like a dimmer switch or changing the gain on a receptor versus just turning it on or off like a light switch. Because as you go into these diseases where you're going to be doing chronic daily maintenance therapy for 20, 30 years,
right?
When you just turn receptors completely on or completely off, they can downregulate, they can be internalized, and then after a while you lose efficacy. So having something that just turns up the gain keeps everything in its normal physiological state.
Right. So somebody with Parkinson's is going to live with Parkinson's, not just for 2 years or 5 years or even 10 years, but potentially 20 or 30 years. So you have to find a medicinal solution that they can utilize over that entire time that essentially doesn't burn out or max out the system.
Right. So for Parkinson's, for example, the standard of care since the 60s has been L-DOPA, various variations of L-DOPA to increase dopamine. But being an agonist, what happens is you get the on-off periods and wearing off effects, and then you develop dyskinesias because you're hitting the receptor so hard and keeping it on for so long. You have all types of negative side effects.
Amazing. And I think that sense that I read from your papers that you're managing and really monitoring all the other ways in which that particular medicinal solution might be negatively affecting, whether it's kidney or, other portions of the brain, that you have to have this incredibly precise way of targeting a specific, location at a specific time with a specific dose for a specific period of time so that that dimmer switch isn't drastically cranked up or drastically cranked down.
That's correct.
Amazing. Just incredible to think about the precision with which you have to actually do that. Tell us a little bit about the ways in which restoring healthy brain function is possible. When you think about the outcome that you're trying to achieve with somebody with Alzheimer's, as you develop this particular set of medical solutions, these particular chemicals that could serve as drugs, what are you shooting for? What's the hope?
Well, the way we think about it a lot is you think about the patient and the patient needs, but a lot of times with severe brain disorders, it's actually harder on the families and the caregivers than the actual patient because they may not really be aware of everything that's going on. So we really think about what can we do to make quality of life better for the patient and their families. So that's kind of the guiding principle. But if you think about so many of these brain disorders, if you have a circuit, just like a circuit in a light switch, right? If you turn it on all the time, it's going to burn out. It's not going to work very well. And if you just potentiate it, if you just modulate function and increase the gain just a little bit, that can actually, in certain cases, not only can you get palliative benefit on cognition, but you can actually start to have disease modification because you're not burning out all the downstream receptors that are just getting, constantly being activated. So a lot of the mechanisms we work on in targets with PAMs, there's data suggesting they're neuroprotective and neurorestorative over time.
Amazing. So you're not simply just trying to ease the progression of a disease like any neurodegenerative disease, but in Alzheimer's or other forms of dementia, you're actually trying to support the brain in terms of the way in which that set of pathways may be sort of assaulted by that neurodegenerative process, that you're trying to keep people in a healthier state for a longer period of time.
Correct.
Amazing.
But for a clinical trial, you have to go for just an improvement in cognition because you can't run a 10-year outcome trial. But with collaborators in Scotland, Andrew Tobin, we did an almost 2-year-long study with one of our compounds in mice, and you could show that you increase lifespan significantly and performance, and so it seems like it's possible.
Incredible. And in terms of cognition, is it about restoration of memory or preservation of memory so that people can remember their loved ones and the match names and faces as people come in the door? Is it remembering things like where they put their keys or whether they just gave somebody, a large chunk out of their savings account? Or is it more on the behavioral side?
Well, it's all those. We'd like to make have people have better memory, short, long-term memory, but it's also a lot of the things we work on are executive function. So you can remember, I put my keys here because I need to go here next. And those kind of things, which, even in schizophrenia, people typically think about just the hallucinations, but there are massive cognitive deficits, which is why even folks that are on atypical antipsychotics, they can't seem to really work. They can't integrate into society. And it's because of the massive cognitive disruptions.
Yeah, making decisions.
They can't, yeah.
Yeah, making key decisions that are wise rather than driven by sort of the more appetitive or emotional driven mechanisms. Incredible. Walk me through the drug discovery process. I think the extent to which I loved in reading your papers, you provide a diagram for each one of those chemical compound variants with just tiny changes in what look like changes in covalent bonds at the top of a ring, or a change in atomic structure, or a change in the way that ring was oriented, whether it was oriented to the left or to the right. So it strikes me, as a person who doesn't know your field, that you run these large number of variants through these successive set of gates to try to determine which of those many, many keys might fit the lock. Is that the right analogy, Craig?
That's really accurate. Yes, I don't know if everyone realizes that a drug discovery program, from start to getting a compound approved by the FDA and launching and actually having it available in a pharmacy, is about a 15-year process.
Incredible.
The work we'll do in the laboratory will typically make maybe 10 to 20,000 molecules, and there's iterative rounds of chemistry, biology, drug metabolism, because it's not just on-target potency and selectivity, but then can you ingest the molecule? Does it dissolve? Does it get into the bloodstream? Does it cross the blood-brain barrier? Does it get to the location in the brain where you need it to be? And does it do so without any kind of toxicological findings? And so
it might be helpful in one regard, but negatively consequential in another. It might actually,
So when you're doing neuroscience, we have to be really cognizant of liver toxicity, because this is long-term cardiovascular toxicity. But, it's typically 2 to 5 years in the laboratory, and then the next 10 are all under the FDA guidance for the investigational new drug enabling studies. Then phase 1, where we're in healthy volunteers, then phase 2, where you're in patients, and then phase 3, where you do a much larger multi-center trial with heterogeneous patients to make sure it works. So it's a long process, and you go from 10 to 20,000 compounds down to 1, and then still, once you're in clinical development, there's about a 95% failure rate, because there's just things you can't predict, idiosyncratic toxin, other kind of things. But if you imagine from a business perspective, if you were building cars or televisions and 95% of your cars got scrapped on your assembly line, it's not a very good business model. It's very, very hard.
Very, very costly in terms of both, obviously, all the things that go into that, including many, many mice, as I read.
A lot of animal models.
A lot of animal models, a lot of reagents, a lot of chemistry, but also a lot of people.
Mm-hmm. So, before we can go into healthy volunteers, we have to do toxicology studies in a variety of different animal species to make sure it's safe before you go into humans. I think it's a really interesting story. I don't know if you know this one, why the FDA got started.
No.
So right after World War I, back then, there were no labels on anything. And a lot of drugs and medications just had cocaine in them and things like that. But after World War I, there was a lot of bacterial outbreaks in the U.S. from people coming back. One company took an antibiotic that wasn't soluble, so they dissolved it in ethylene glycol, which is antifreeze, and added raspberry syrup to it, and directly went and gave it to thousands of children. And about 450 of them died immediately from renal toxicity, kidney failure.
Okay, so that was a medical advance, quote unquote, generated by a corporate entity that was completely unregulated. And
there was no regulation at all. So then they started the Food, Drug, Cosmetic Act, which said you have to first test this in an animal. And when they test that same formulation in animals, the animals all died. So had they done this first, you would have saved all these children. So then that led to the FDA and drug safety and drug monitoring. And so now before something moves into the clinic, it is very, very well vetted.
Yeah. And I love the layering of all of those processes. Could you make the chemical compound at scale, that you started with? Is this even something that is replicable? Is this something that we could actually manufacture?
That's another problem. So in the laboratory, you're making milligram quantities, and maybe you'll make a gram at the end of the day, which isn't really that much material. There's 2 grams of sugar in a Coke, so make about a gram of material. But then when you go into the clinic, you're making 10 kilograms, and so you have to develop entire process plants that just make your drug. And a lot of times, there can be failures, there can be impurities you can't get out. The cost of intermediates can go up. There's all kinds of issues. So molecules can fail in so many different places along that continuum.
So fascinating. So you have this, chemical set of constraints. Can you even make the stuff you're trying to test, right? Mm-hmm. Can you make it well? Is it pure? Does it have other stuff hanging on to the edges of it, essentially, that might be toxic? And then is it essentially targeting the actual regions, the brain regions that you hope through both mouse models and rat models, other animal models, to even see that it is achieving even close to what you're hoping to achieve? I love that you even include behavioral models, that you had this way in which you would try to essentially instantiate schizophrenia-like symptoms or Alzheimer's-like symptoms in mice and see whether the candidate chemistry that you were developing was demonstrating that kind of behavioral impact. Can you say a little bit about how the worlds of your labs work?
Sure.
Obviously, one side, the beaker and reagent side, versus another side of your lab, the animal model side, must be an incredibly vibrant, active group of people, but not everybody's doing the same thing.
Sure. Drug discovery is probably the greatest single example of big team science. So you have people who are subject matter experts in different areas, but then they all collaborate and integrate and just work seamlessly across the task. So the chemists make the small molecules. They synthesize them, purify them, characterize them. Then the molecular pharmacologists test them in cell-based assays. Are we potentiating the receptor we want to, or inhibiting the receptor we want to? And then they go into our drug metabolism group, which does in vitro work in cells first to see, is the compound highly bound to plasma proteins or not? Does it have stability in rat plasma? Does it get into the brain? We do in vivo studies. What's the pharmacokinetics? Is it orally bioavailable? Can you take the pill and it get to where you want it to go? And then on campus with Dr. Carrie Jones, she does all of our neurobehavior. And so for every program we work on, we have a primary pharmacodynamic assay. We could be doing a pharmacological challenge. We could be inducing a schizophrenic-like deficit or a Parkinsonian-like deficit with a drug, and then we try to reverse that. Or in a lot of cases, we have genetic mice. They actually carry human mutations that we know are associated with the disease, and we see if our molecules can reverse those phenotypes.
Incredible.
And then it's just a constant feedback loop.
Right. So what you learned at that later stage, you then carry back to your chemists to say, okay, we actually need to utilize that other variant of that compound because this is yielding not quite what we hoped.
And that's why, like you see in these tables, sometimes the changes in the molecule are very, very subtle because we're just trying to fix a drug metabolism liability, a solubility liability, trying to decrease exposure. And a lot of times the changes are really minute, not always really big changes to get where you need to go.
Amazing. And I guess what it leads me to realize is this incredible work that you're doing to keep me safe, right? That, when I, or anybody that I love, or anybody in our world, or anybody in our community takes a medicine that we purchase at the pharmacy, that there's 10 to 15 to 20 to 30 years of all of that work going to ensure that that little pill is not just manufactured correctly, but that those active ingredients, those active molecules have been tested against so many different challenges that have utilized so many different hands on deck, so to speak, to ensure its safety. How do you maintain that level of clarity and commitment and energy, knowing that you're not going to see, a result for 95% of what you're doing, and that it's going to be years?
It's just the challenge of doing it. Yeah. I think especially working in brain health, it's the hardest, right? These are daily maintenance therapies for 20, 30 years. Getting compounds that are orally, can be taken orally and get to the brain is probably one of the hardest things to do. Oncology drugs are acute, bacterial acute, viral acute, so this is really a challenge. But it's, I I think everyone that works at the Warren Center just loves the challenge of it. And there's always the chance that your compound's going to make it. So, it's just a different mindset.
Does it have that sort of wonderful Vanderbilt SEC, I love a challenge and I'm particularly competitive? Are you trying to beat another lab? Is there a sense of, sort of race to the finish? Or
So I think one of the greatest things about doing what we're doing here at Vanderbilt versus when I was, say, in industry at Merck, when I was there, if we were working on a program and we did not develop the drug, we failed. And it was all viewed as a massive failure. Here, we have so many examples of where we started doing basic science around targets, developed tool compounds, validated the target in animal models. And that led other pharmaceutical companies to start programs, and they all cite our papers and our patents. And that's actually how we got some of our deals. Companies have come to work with us because of our basic science papers on things. But like some of the programs that are in the clinic right now, we actually beat the big pharma because we invested more time in really understanding all the little caveats around the target and what's necessary. So
I love that.
A lot of things like that, but it's great because at the end of the day too, is if the science we do here at Vanderbilt inspires other companies to start the same programs, whether we get the drug out at the end of the day, it's a Vanderbilt drug, or if it's a Merck or a Pfizer drug, but the patients benefit.
It's awesome. I mean, I love...
the impact of that is just amazing.
Right. I love the impact. And I really love the idea that early stage failure, quote unquote, could either be viewed as failure or it could be viewed as catalyst, right? It could be viewed as early stage breaking open of something that we didn't know or that is disconfirming and that inspires actually more of us to jump in and try to tackle it, right?
Sure. And I think the way we approach things too, or the way I always approach things is a little different. There are so many possible targets we could work on, so many. And with BioVU and other genetic, we're getting so much input on genetic dysfunction that's leading to certain diseases, which gives ideas for targets to work on. And so the way I think about it in the lab is, we want to kill the compound. We want to kill the series. You want to kill the target. You want to kill the mechanism. Because, if we can do that as well, it's great basic science. It lets people know where not to go and allows us as a collaborative, community to focus on the targets that really could impact patients.
Cool. So when you say kill the compound, you mean basically scratch it off the list.
Let's find what's wrong with it. Where is it deficient? Let's do every experiment we can to try to knock the compound out of the running. And if you do everything you possibly can and you can't do that, that's how we get some of these molecules that then moved into human testing, because we did everything we could to prevent it from going forward, and it still made it. Wow.
So that's just to circle back, then, talk about keeping us all safe. The med that we're taking is the winner after all those other candidate chemical compounds have been knocked off. So it wasn't like, the first one out of the blocks.
Oh, no.
It's the absolute reigning winner after so many forced kind of comparisons and challenges.
And I think if you look over the last 30 years, there's been such a great interaction between industry, academia, and the FDA. And every time they start to identify drugs that cause liver toxicity or cardiovascular toxicity, industry and academics partner together. They figure out what that cause is, and now that's part of the testing cascade. So you don't see drugs being recalled anymore for drug-induced liver injury or for cardiovascular or QT prolongation, because that communication loop just gets amplified, and everybody goes and addresses the problem and figures it out. And then,
awesome.
There could be something else looming we don't know about yet, but we can definitely eliminate all the known knowns and bring safe drugs forward.
It's stitched into the process instead of being something that we hope doesn't happen down the line. I was thinking a lot about that, and I just want to first note that synergy between industry and the federal government and research labs like yours at Vanderbilt. When I look at this paper, Craig, I notice right under the title, there are 26 authors, right? So it's not you claiming, ta-da, a new drug, right? It's you and 25 other people. Tell me about this amazing team.
Well, yeah. So this is, again, like I mentioned, this is big team science. So anytime you work on a program like this M1 positive allosteric modulator program for Alzheimer's and schizophrenia, you've got a team of 6-7 medicinal chemists working on it. You've got 2 molecular pharmacologists running the assays. You've got a team of 3 or 4 drug metabolism scientists, 2 or 3 behavioral pharmacologists. And then we have the toxicologist, because with this program, This was one where the Warren family, the first gift they gave us was to provide the funding to take this compound through IND-enabling toxicology, which then poised us with Paul Newhouse to get Alzheimer's funding to go into the phase 1, which was run here at Vanderbilt.
Amazing.
So it takes everybody working together, and I mean the integration, the communication. It's like nothing else.
It's great. And is that largely mid-career scientists? Is that junior scientists? Is that undergraduates and graduate students? Is that postdocs? Who are those toxicologists and those behavioral pharmacologists? What stage of career are they?
So these are usually, so this would be like Dr. Carrie Jones and folks in her lab. So they're more senior scientists because this is really tricky stuff.
Yeah.
When a program becomes a real drug discovery project where we're developing IP and really going toward a clinical candidate, then it's really just the staff scientists in the Warren Center. So these could be junior medicinal chemists, senior medicinal chemists, same thing on the pharmacology side. In terms of toxicologists, we typically go to CROs for that because we don't have internal toxicologists.
Sorry, what's a CRO?
Contract research organization.
Got it.
But in the earliest genesis of all these programs where we're trying to develop cell lines and make the first tool molecules and target validation, those have always been done on R01s or other types of federal funding or foundation grants. And this foundation of all of this is done with grad students, postdocs, undergrads. And think about it too, for training for students to be able to walk through, chemistry, pharmacology, drug metabolism, behavior, and see something that they started on going through all those iterations and validating a target. It's been really phenomenal training for those students. And now they're leaders in industry and academics that have
super inspiring, right? They go on to those incredibly important next steps in their
because they're not just, you can get really niched. I could just say I'm just a medicinal chemist and I don't do anything else. But the exposure you get and the access to all those other things, people start thinking differently, asking questions, and they become really holistic drug discovery scientists, not just a single discipline.
Makes total sense. And I've loved walking through your lab. I've loved seeing the incredible energy. It's very calm, but it's also, and very focused, but there's also very high energy. There's a lot going on.
There's a lot going on.
It's really impressive. And I think a lot about the way in which you're contributing not only to the nation's health, but to the nation's economic strength, right? That those 26 people are supported by probably another 40 lab technicians and everybody from the building custodian to the real estate person that rented you the research space, right? That I think about how you are contributing, whether it's in Franklin, Tennessee, or for the state of Tennessee or for the region, to this sort of massive endeavor, which is actually a key strength of our economic competitiveness worldwide.
It really is. I mean, we're creating a lot of economic growth in the area, and the Warren Center is really probably the only biotech in Nashville, the Nashville area, and it functions like a biotech.
Yeah, and clear from the list, I loved that you gave due credit for each of your authors in terms of who they are, and in the back of the article, a couple of them are from pharmaceutical firms, and the funding is partnered with Acadia Pharmaceutical. That level of industry partnership, is that typical of the work that you do as a scientist that's discovering new medical breakthroughs? Yeah,
so for us in the Warren Center, we can get federal funding and foundation funding to do all the discovery and get compounds to the point of where they're ready to go into IND-enabling studies. But then those costs just are exponentially grow. So far, we've had 37 collaborations with big pharma and biotech, and they typically will come in and they'll sponsor research to get to the clinical candidate and backup compounds. Then they carry the burden for the IND-enabling studies and the clinical development.
Got it.
So right now we have 4 compounds in clinical development. A spin-out company we had, Apello, they just got their next funding round and they're going to be going into phase 2. So our first efficacy trial in actual patients. The other 3 are phase 1s in healthy volunteers, but there's 6 other programs right now in IND-enabling studies that should be in the clinic.
And sorry, just slow down so that everybody, including me, IND-enabling means?
Oh, sure. So once you have a compound that leaves the laboratory, before you can go into healthy volunteers, we have to do what's called investigational new drug application. And so there's toxicology studies we have to do. And this is where, again, based on what happened with those children in the 1920s, we have to thoroughly vet our molecules for toxicology and safety in multiple preclinical species, very rigorous. those results go to the FDA who review them, and they can give you permission to proceed into a phase 1 trial, or they can give you a clinical hold, or they can just deny you. They don't feel the safety is where it needs to be.
So it may be 12 years of work, and then you hit this key gate, this key moment in the finding of the key into the lock.
It's
if that tumbler can turn or not.
It's the longest 30 days of your life. So once you submit the IND application to the FDA, they have 30 days to review it and respond and either say you can proceed to the clinic or not. And that's a very long month.
Wow. And does the lab, toast with a glass of champagne when the answer is yes? How do you?
We have done that. That's a big accomplishment.
I mean, it must just feel incredibly gratifying after those intense months and years.
It is, because when you're in industry, that's just, you expect to do this kind of work all the time. And that kind of level of things working out is just expected. When we first started the center, we really were thinking more about just validating targets and not really translating things all the way into the clinic. And the fact that now, the center is delivering on a pipeline, and there's all this momentum and things just keep progressing and going to the next milestone. It's amazing, because we honestly didn't think we would have gotten to where we are. So it's just really fantastic.
Incredible. I think many of us don't realize or can't necessarily access the image that you are both, bench scientist and CEO, right? That you are both discovering new compounds and running this massive, huge enterprise that has all these people that are contributing not just to science, but to the economy. It's just a kind of profound sort of double role that I can now understand. Do you have a favorite role? Do you like being the CEO more than the scientist, or the scientist more than the CEO?
Doing this in academics is interesting, because you're like a CEO, a CSO, a CFO, you're the IP attorney. You're doing all these different things.
Cleaning the kitchen sink when
trying to negotiate contracts with people, reviewing terms. I mean, you're doing so many things, right? And it's all essential, and it has to be done to keep progress moving and getting programs developed. But there's something about when you were a bench chemist and you designed a molecule, you made it, and then that became the compound that moved forward. There was just something really rewarding about that. And it's harder when you take yourself out of the laboratory and you're doing all the higher level things that are essential.
Yeah.
But you don't have that tangible reward of, that's my compound.
Further from the bench, or further from the actual testing of the animal model, et cetera. Yeah. Now feels like a good time for us to take a quick break. We'll be right back with the next segment on pain management. So welcome back, Craig. It's been so fascinating to talk about all your multiple roles and the incredible power of medicinal chemistry to address issues like Alzheimer's and Parkinson's. I'd love to turn right now to some of your recently published groundbreaking work on discovering drugs that might dramatically change the way we treat pain. As we've seen from the opioid crisis in this country, we desperately need better alternatives to treating pain. Can you tell me a little bit about your work in this area?
Sure. So we've been working in pain for quite some time. A lot of our targets cross over across multiple therapeutic areas, but with Ono Pharmaceuticals, for example, we've had a 12-year collaboration with them, and it's always about non-opioid analgesia. After all the opioid crisis, we're always looking for non-opioid medications. So with Ono, we've been working on ion channels. It's a different type of receptor that conducts potassium ions across membranes. And so we developed the first TREC1 inhibitors or activators, and they're very efficacious in multiple models of pain, and they don't have addictive liability, unlike opiates.
And as I read the paper, you screened 20,000 candidates to find those TREC1 and TREC2. And
to find one lead compound.
To find one lead compound.
Only one lead compound.
You screened 20,000 other candidates. Mm-hmm. Amazing. Okay, so how long did it take to screen 20,000 camp?
Oh, the screens are fairly high, a few months.
Yeah. Okay. So a few months, you narrow it down. And then that paper was sort of like a horse race, as I could read it at least, that you're really challenging one candidate against another to try to understand which is more efficacious.
Mm-hmm. So it's always going to be subtle things with potency, efficacy, exposure in the brain, or can you get efficacy in pain without having to get into the brain? The root, the dorsal horn, there's a lot of pain sensory neurons there. But we also had some large grants from NIDA to work on different pain targets. One of our mGlu5 NAMs was a HEAL grant. Compounds efficacious in multiple types of pain. Mm-hmm. There's lots of different forms of pain, and that's looking really good. We're in discussions to license that program now. But again, it's always, you have to do all the addiction liabilities and abuse liability studies to make sure that you're not just making an opiate by another name.
There you go.
So we have to do all that work up front.
And is the screening as rigorous or even more rigorous than what you just described for the previous rounds in terms of the IND?
Yeah, it's much more challenging. Again, pain medications have to really be exquisitely safe and clean and non-habit forming and those kind of things.
I love that phrase.
For other things, you don't have to. For other targets, you don't have the same liabilities.
Yeah. And then, you mentioned that NIDA supports that. That's the National Institute of Drug Abuse. Good. Okay. So I know my federal agencies just barely. That's been in the news a lot because of the National Institutes of Health experiencing such dramatic changes in sort of their ability to distribute funding. What does that mean when the National Institutes of Health has to curtail or limit its ability to disperse funds to labs like yours? Yeah,
it's really, not to be overly dramatic, but it's very crippling, because if you look at the history of the center, 95% of the programs we've worked on started with federal funded R01s and U01s and things of that nature.
And R01s, just for the folks listening, that's a starter grant, right?
That's a basic 5-year starter grant. You have an idea, a concept. In our case, maybe it was we think we can develop selective M1 positive allosteric modulators, and we think they're going to be good to restore cognition. And that gets funded, and then that gives us the money to develop the cell lines, make those early tool molecules, do the target validation.
And tell me just a little bit, while we're on that point, that early funding is a risk you take as a scientist, and it's a risk certainly that the federal government takes in terms of trying out a new idea. Is that a risk that a pharmaceutical firm would ordinarily take because they're so needing to meet the bottom line, right? As you mentioned earlier, they've got so much pressure that they're, as you described it, they're likely to interpret that early stage work as super high risky and potentially as too high risk for failure. Is that right?
That is spot on. So with federal funding, some of these grants have a 5-year term. So you have 5 years to spend really getting into the weeds of all the nuances of these targets. When I worked in industry, you would be assigned a program, and within 18 months to 2 years, you had to have the clinical candidate. So you threw an assay together pretty quickly that may or may not have been the best. And you drove on that because you had to hit the milestone to get the metrics, but you didn't really understand necessarily the target as well as you should.
Wow.
And that's why in several of our programs, we've been able to outpace big pharma because we just spent a lot more time understanding it.
Amazing.
And NIH enabled all that.
Got it. So the National Institutes of Health, that investment, that infusion of federal funds, essentially, as I hear you say it, allowed you to do better science that actually ended up being more effective and more efficient in the long run. Much more thoughtful science, and having the time to just ask questions. What are the liabilities of this mechanism? How can we address those? Maybe we need to go into move from cells to native tissues and do electrophysiology. Maybe we need to do different types of animal models. Maybe we need to take compounds with very different profiles and put them in animals. And what does it look like? And it's just things in industry, because it's just, you got shareholders you're accountable to, and you've got candidate metrics every year that management has to hit for their position. So it's academics is the only one you can, and you're not really being leisurely, but you do have the benefit of time. Yeah. And your shareholders, it sounds like your shareholders are the American people. That's true. Yeah. I think it's just so interesting to think about, and so important to think about, not simply interesting intellectually, but deeply important in terms of the way the machine works to lead to our ability to outpace other nations around the globe, and how we inspire both the creativity and the competitiveness that your lab really serves as such a great model for. You're super creative. You're going after majorly difficult diseases like Alzheimer's and Parkinson's, but you're also incredibly thorough. I love how you manage that. And, the extent to which you're doing that because you both embrace partnership with pharmaceutical firms, and you capitalize and use so effectively that early stage federal investment.
And I think another really important point, doing this type of work in a university setting, all of our scientists are here. We're all in the same building. We talk daily. We're integrated. What you've seen in industry over the last 20 years is they outsource more and more work to foreign countries. They'll outsource chemistry, they'll outsource biology, they'll outsource behavior and drug metabolism. And they'll have one or two scientists at the company that are directing all these offshore work.
I never thought of offshore.
And there's just not
work.
Communication.
Yeah.
I mean, you don't have the communication, and you don't have leadership in all these areas talking daily, discussing the data and the implications of it and how to modify it and what we need to, how we need to pivot. And while they save money in the short term, it takes far longer to get to a clinical candidate than if they did it all internally. So if you think of a drug's going to make a couple $1 billion a year and it took you a few extra years to get there, you didn't save billions on the discovery.
Yeah.
But universities are just poised to be more successful just because we're in-house integrated teams.
Fascinating. I mean, I never thought about us as in-house, right? That we don't offshore our scientific discovery. We do all that work where you, Craig Lindsley, can walk the halls and check in on what's going on in that particular moment where things might be going a little sideways or things need to get
It's basically the
straightened out.
pharmaceutical industry in the 60s and 70s when they're probably the most successful. I mean, we're still taking drugs today developed in the 60s and 70s because of that tight-knit integration and close working interaction that the industry had, and they've moved away from that.
Incredible. So in that way that you are then partnering with pharmaceuticals, how does that relationship work? Because, obviously that's a huge strength, I think, of American scientific enterprise, that we figure out how to accelerate the impact of the work that our scientists do by getting it out into the market, into the real world, by partnering with corporations. Tell me a little bit about that partnership.
So it's been a great academic, industrial kind of balance. So all the papers on the early proof of concept studies are kind of the billboards for people to want to come work with us. And then all of our, the industrial collaborations come in many different flavors. For most of them, they fund us to do all the research here, and we agree on what a candidate would look like, the profile. And when we achieve that, then they take it and do the development. We've had with some other large pharmas, there's a research team there working on it as well. So the chemists from both sites have target meetings and interact all the time. Same thing for all the other disciplines. And then we jointly develop the IP and get the compounds into the clinic. So it really kind of depends.
Yeah.
But what's been so great about it is, like, not just to pick on Merck, but when I worked at Merck, there was the Merck way of doing everything. It was. This is how we do it. This is the workflow. This is what you do. Having worked now with 37 different organizations, you kind of see how every company, based on their past experience, does things a little differently.
Wow. So you work with 37 different corporate entities.
Mm-hmm.
Amazing.
And so now our workflow, like our internal Warren Center workflow, takes the best from all these different people. We've integrated it. So it doesn't look like anything I've done before. And it's the best of all worlds. And if you're working in a company for 30 years, you have no idea what the guy down the street is doing. We've got to see it all. We got to see under the hood of everybody and take all the best elements. And so when we start doing our early stage projects now, we're using these workflows that are really the best of what you'd see in pharma. And so we're able to get to compounds pretty quickly that then garner interest from other companies and so on and so forth.
Amazing. So you serve as this interesting hybridizing of the methods of science, not just of the actual, compounds that you discover, that you're sharing processes of how to either manufacture or test those candidates across all of those different corporate entities and across those different platforms. Is that something that's explicit? Like, hi, I'm a new pharmaceutical company and I'd like my people to train with you, Craig. I'd like my people to be embedded in your research lab. And, 2 years from now, I'd like some of your people to be embedded in my corporate entity. Is there that kind of hybridizing?
We've done that as well. So with Ono, that collaboration started in 2012. And the original collaboration was just they sent scientists here to work in the lab and see how we do things. Then it became a large sponsored research program. And then during the course of that, they would usually send 1 or 2 chemists to work in our labs and a biologist to work on the labs with Jared Denton on campus. And they would even send behavioral pharmacologists. So it was great, and also just the whole culture, working with people all around the world, all across Europe and Asia. It's been fascinating, just the cultural exchange as well as the scientific exchange.
Sure.
It's just fantastic.
What are you having for lunch? When awesome. Do you work on Saturdays? Right, I'm sure. Right, and I guess that's really, I think, also a surprise to me. Right, that I think about executive education, for example, in business. That, corporate entities send some of their mid-career executives, their leaders, to a university like ours to learn more about how to run the business effectively. But I don't often think about scientists in the, pharmaceutical industry or in the drug discovery industry outside of universities as also sending their mid-career team to learn that there is that exchange in terms of training, not just in terms of intellectual property. Really exciting.
And you just touched on intellectual property too. It's different companies in different regions of the world approach patent filings very differently. And so that's also been a real educational process to see how everyone just approaches it so fundamentally differently.
Right. How do we claim credit? How do we
win? How
there's just how do we earn? Yeah.
It's really interesting.
Fascinating. And did you know you were always, when you were 6, sitting at the kitchen table, I have no idea what you had for breakfast, but when you were sitting there, prior to heading off to school, first grade, did you know that you were going to be, a leader in drug discovery? What was the path that you followed to get here, Craig?
Let's see, 6 years old, Star Wars came out. I think I wanted to be Luke Skywalker, but that didn't work out.
All right. So you're not an astronaut.
No, when I started college, I started off as an English major.
Wow. For all our English majors.
Yeah. And I ended up taking chemistry classes and took organic chemistry, and that just all made sense.
Wow.
Ended up getting a minor in English, which has been so helpful because, I think one of the biggest things with scientists is being able to communicate orally and written. And all we do is write papers and patents and grants. And if you're not a good writer and you can't tell a compelling story and intertwine the data in the story,
right. Scientists,
you're not going to get
writers.
funded. So
absolutely.
I think it was a very good call.
That's great. And did you have a chance to be in, the sort of standard track of graduate school and then a postdoc in somebody else's lab, and then you went to Merck from there? Got it. Yeah.
PhD, postdoc, and then industry for 10 years, and then came here.
And how do you explain to your family or your community what you do? Do they get it?
Yeah, I think they get it at a very high level. They think we're trying to make fundamentally new drugs for various, brain disorders and for brain health. And it's hard and it's challenging. And, just like with, everyone knows someone that the family with cancer. It's amazing as you start talking to more people. And I think as the population is getting older, everyone I know has someone in their family suffering from Alzheimer's or Parkinson's.
Absolutely.
And actually schizophrenia, which is surprising to me because it's somewhat rare, but the number of times people say, well, my sister or my mother, and I go, wow, it's surprising.
Yeah. Well, and I think some of it probably is because of the way in which we're getting better at diagnosing, right? So perhaps before we said, oh, somebody's aging and they're getting forgetful or they're becoming difficult to manage. But now we're clearer about that being a neurodegenerative disease and that's something that is happening in their brains that's atypical and needs care.
And I think finally the stigma is not as great as it used to be. And one thing we talk about all the time is brain health and brain disorders. We're not really talking about mental health because there's the negative connotations of that.
Yes.
And I think more and more people, even adolescents, are asking for help and telling people when they're struggling or having problems. So we're finding it out sooner and being able to hopefully have interventions earlier.
And I love that your lab is committed to that, to some of that communication effort, whether it's inspired by your English interests or in your prior experience just communicating to a broader community, that brain health is, a key area that people deserve to be able to pursue the health of their cognitive function, their emotional function, their sleep, their well-being.
Well, I think it's helped a lot too when you have folks like Michael J. Fox being so public about it. It's really helped, and then for other disorders and diseases as well, more and more people are coming out and talking about things, and and there's sympathy for it now, which I think is so important because for a while, if you had a brain disorder, people treated you differently and looked down on you, and you would not look down on somebody that had cancer or something. It's just really
not a source of shame, but instead a source of seeking help. Absolutely. Amazing conversation so far, Craig. I'm so grateful for being able to kind of have you explain so much of the intricacies of the work that you do. If there's one message that you wanted our listeners to take away, what would it be?
Well, I think where we are right now, and not trying to be political, but just in general where we are right now and the damage that's being done to universities and the NIH, it's going to be a big detriment to discovery and developing new therapeutics in the future. We're going to have, with things that are going on, I really worry that you're going to have a gap in people coming in that can do the work we're doing now.
Yeah.
That's a big concern for me.
And can you say just a little bit more about kind of the ripple effects of that concern? If we didn't do work for the next 2 years in your lab, if your lab had to take a pause, what would be the downstream effects, whether it's for Franklin, Tennessee, or for the scientists in your lab, or for people like me who have a friend or a family member with Alzheimer's or another neurodegenerative disease?
Well, obviously, any kind of pause is going to delay things moving forward. And then you're also burning patent life during the course of that time. And then that makes the products less attractive to companies because they've lost less of their exclusivity time. It's just...
So wait, that's interesting.
Delays are exponential.
Delays are exponential. And you just described delays in the business sector, that the intellectual property doesn't hold its value in perpetuity. It's
no, you get a set time you get 22 years of patent life when you file a case. And if it sits idle for years, you don't get that time back. And then, industry can view those as damaged goods because, well, now, if it does take 15 years to get to market, we're hoping to have 7 years of exclusive sales to recoup for all the programs that failed. And so, delays, multi-year delays, the value of that asset dropped, no matter how good the molecule is, no matter how impactful it could be, there's not that financial gain at the end. So they may not license it and develop it.
Wow. And what about loss of optimism or belief in doing science? Do you think
that's even more concerning? Because as students, like you've seen, a lot of universities are rescinding graduate student offers that have already been made. They're not accepting students this year. And then at NIH in the intramural program, one of my former students, on a Saturday, they got an email that their tenure track jobs were terminated and they had to be out by Sunday. And, so you're just having a whole generation of people that are very talented and very capable now looking to leave science. I don't know how you're ever going to rebuild the intramural program at the federal government because there's no stability. And there's little stability already in industry. And there's, you're going to face it. Why would people want to go into science? It's
right.
So
the thing
hard.
You just described is so hard.
And the training to get there takes so long.
Right.
And then if you're in a position where, any one administration can just change everything and eliminate all, it's really scary. It's scary for the world.
I hear you. And, I think the flip side is that I see you and your team figuring out a way to really maximize every dollar invested, whether that's a federal dollar invested 2 years ago or a federal dollar invested yesterday or a corporate dollar or a philanthropic dollar, that you're really pushing hard on the value of every one of those dollars invested. And, I remain hopeful that we will find means of trying to secure as much of that support as we can, because it is so incredibly valuable. I'm so deeply appreciative of the kind of commitment that you make, but that all of those 26 people make, that all of those other 40 people that support those 26 people, right? That amazing group continues to really thrive and flourish.
Well, thank you. I think, the folks at the Warren Center are just some of the best. They're the best scientists. They're the most dedicated people you're ever going to meet.
Dr. Lindsley, this has been such a fascinating conversation. And for those who listen, believing in the importance of neuroscience research is really clear, I think, from this conversation. What actions would you want us all to take as we move forward?
Support scientists, support the folks that are out there trying to make a difference and impact, the lives of your family members. Be kind. People are struggling. Brain health is issues are growing exponentially, and people need care and compassion, and hopefully there'll be new therapeutics on the market soon that will really change their quality of life and their families.
I love that. I love that, something as dry as chemistry leads us to care and compassion.
Yeah.
Really inspiring, really fascinating, Craig. Thank you so much for taking the time. I really want to applaud you for not only protecting our nation's health, but really serving as a galvanizing force in American enterprise. And it's just really exciting to see where the Warren Center and where your own leadership will go. Thanks to our listeners for tuning in. Remember to stay curious and keep exploring new ideas. We'll be back next time with another guest who's pushing the boundaries of discovery. Until then, I'm Cybele Raver, and this is Quantum Potential.
The Quantum Potential podcast comes from Vanderbilt University. Your host is C. Cybele Raver. Thank you to our guest, Craig Lindsley. Production by Matriarch Digital Media. For more information about Quantum Potential, go to vanderbilt.edu/quantum potential. Stay curious.